everyone, I am Bhavna Rai and in today's video, I am going to talk about secondary growth in a dicot stem. So if we talk about secondary growth, it is a characteristic feature of stem and roots in a dicot plants and gymnosperm. Secondary growth is absent in leaves and in the monocot plants. It is not a feature that occurs in a monocot plants with few exceptions of plants like date palm and coconut where we can observe an exceptional secondary growth as you can see in the examples. So what is basically secondary growth? It increases the girth of the plant body or width of the plant body. Primary growth in a plant increases the length of the plant body and it is a feature of all the types of plants and all the organs present in the plants. But if we talk about secondary growth, it is only observed in the dicot stem and root and in the gymnosperm stems and roots in a general case. So here we are discussing about the secondary growth in the dicot stem. So we are going to talk about the stem of a dicot plant that shows secondary growth. As the name suggests, secondary growth helps in the formation of secondary structures inside a plant body. During primary growth, we get to see the formation of primary tissues, primary organs, but during secondary growth, we get to see the formation of secondary tissues that leads to increase in width of the plant, horizontal increase in the plant. So here we are taking a case of a dicot stem. This is a plant body. So we have taken a horizontal section, the transverse section of the plant. So we cut it and we study the horizontal section, observing different layer of cells in the dicot stem anatomy. If I talk about dicot stem anatomy, the outermost layer of cell is epidermis. Below that layer of cell, there is a hypodermis. Below hypodermis, we get to see the endodermis and a layer of cell called as pericycle. In that layer of cells, the pericycle surrounds vascular bundles inside the stem. Vascular bundles means xylem and phloem. What is the function of these tissues first of all? We all know that phloem and xylem are known as conductive tissues of the plant. They conduct water, minerals, food, hormones, etc. across the different regions of plant body. So here, as the plant grows towards maturity, along with primary tissues, it needs some secondary tissues as well for more conduction purpose and strength to the plant. So the vascular bundles are phloem and xylem. Now you all can observe there is a ring arrangement of the vascular bundles within the dissection, within the cross section of the stem. Between the vascular bundles, what we can observe, there are some cells which are known as medullary rays. Medullary rays is a tissue that is made up of parenchymatous cells. So these are primary permanent tissues. These are primary permanent tissues. Between xylem and phloem, there is a pink color band of cells if you all can observe. This band of cells is called as cambium, intravesicular cambium. Cambium means a patch of meristematic cells. So what is the difference between meristematic and permanent cells? Permanent cells do not divide at all. But meristematic cells, their work in the plant system is to divide and increase the number of cells within the plant body. So for any type of growth in plant, whether it is a primary growth or it is a secondary growth, we need these meristematic tissues. So here we are going to see that how this meristematic tissue, the layer of cell, is going to help for the secondary growth. So this is called as intrafascicular cambium, intra, within the phloem and xylem. In this diagram, we all can observe that the intrafascicular cambium has joined with some tissue and forms the vascular cambium. So actually, what is this interfascicular cambium? In the last slide, as we have seen medullary rays, the primary permanent cells, those primary permanent cells undergo the process of de-differentiation. The primary permanent tissues undergo the process of de-differentiation to form secondary meristematic tissue. That secondary meristematic tissue is the interfascicular cambium, that is between the vascular bundles. So intrafascicular cambium plus interfascicular cambium, they join together to form a continuous ring that is called as vascular cambium. So here we have to note one point, that vascular cambium is both primary as well as secondary in origin. Why both primary and secondary? Because primary meristem is the intrafascicular cambium and secondary meristem is the interfascicular cambium. As the name suggests, it is a vascular cambium. So definitely what it is going to do here, it is going to form secondary vascular tissues. Secondary vascular tissues means secondary xylem and secondary phloem. So as you all can see in the picture, the outer portion of the vascular bundle, the green color part, is phloem. The inner portion, the yellow color part, is xylem. In a similar manner, vascular cambium towards inside, it will form secondary xylem, and towards outside, it will form secondary phloem. As we all can observe, both the tissues in this diagram. Secondary xylem towards the inner side and secondary phloem towards the outer side, formed by the band of meristematic cells, which is called as vascular cambium. Now, secondary xylem, you can see that the amount of secondary xylem is more as compared to the amount of secondary phloem. What is the reason to this? The reason is that the activity of vascular cambium is more on inner side. The activity of vascular cambium is more on the inner side as compared to that on the outer side. Similar, in the diagram, you can locate the primary phloem and primary xylem relating to the previous diagram. In the previous diagram, we can only see the primary xylem and primary phloem. But now, due to the activity of vascular cambium, we have secondary vascular tissues, secondary xylem and secondary phloem. So now we all can think of a situation that due to formation of the extravascular tissues, the girth of the plant will definitely increase. The diameter of the plant will definitely increase in this case. That leads to secondary growth or increase in the width of the plant. So the process is continuously taking place, formation of secondary phloem and secondary xylem. And in this diagram also, the same thing can be observed, that the activity of vascular cambium is more on the inner side as compared to the outer side. So this secondary xylem is only called as wood. So when we grow a plant, when the plant is young, it has green stem. When the plant grows old, it has a very hard tissue that is called as wood. Wood is formed from the xylem due to the secondary growth in xylem. Now, depending on the activity of cambium, there are two types of wood also, spring wood and autumn wood. due to the differential activity of vascular cambium in spring season and autumn season, the woods are also different. During spring season, the activity of cambium is high. That leads to formation of broad xylem elements. The vessels and tracheids have a broad lumen, right? An idea we can take from here that spring season is going to approach summer. At that time, plant needs more water. So that is why the xylem elements have broader lumen. And in case of autumn season, the activity of cambium is lower. That is why the xylem elements are narrow. They are small and narrow, formed during the autumn season. So the spring wood has less density and is lighter in color, whereas the autumn wood has a high density and it is dark in color. So how to differentiate between spring wood and autumn wood? So spring wood will be light in color and autumn wood will be dark in color. So these light and dark bands of wood formed across 1 year are called as annual rings. These are the secondary xylem. In 1 year, secondary growth leads to formation of a dark band of autumn wood and a light band of spring wood. So these autumn wood and spring wood are also called as annual rings. The annual rings are used to estimate the age of the tree. They are also used to estimate the age of the tree. So what we observed in this video, that in stem, when secondary growth occurs, there is a formation of secondary tissues like secondary xylem and secondary phloem. Also, secondary xylem is the wood that is of two types, autumn wood and spring wood. Why the woods are of two types? Because of differential activity of vascular cambium in spring season and autumn season. In spring, the activity is high. The second thing which we can observe here is that the activity of vascular cambium is more towards inside. That leads to formation of more amount of secondary xylem as compared to the secondary phloem. So I hope you understood it well. Thank you all of you.